Last chapter, growth and development. Now, we talked about growth and development as one of the characteristics of living organisms. We said growth is the irreversible increase in size of an organism. For unicellular organisms, growth involves an increase in the size of the cell. But in multicellular organisms, it involves an increase in the size and number of cells. So growth can be measured quantitatively. On its part, development is the qualitative aspect of growth and involves differentiation and specialization of cells. These cells are then grouped into tissues to perform specific functions. So essentially, it is not possible to measure the aspects of development quantitatively. Measurement of growth. At the cellular level, growth refers to an increase in the amount of protoplasm or cytoplasm, which is difficult to measure. Growth is measured by different parameters such as size, area, length, and number. For example, watermelon cells increase up to 350,000 times, and so growth here is measured in size. For a leaf, growth is measured as an increase in surface area. The growth of a pollen tube is measured in length. When growth is measured per unit time, it is called growth rate. If we consider an increase in cell number as growth, it can be the result of arithmetic or geometric growth. Arithmetic growth refers to the division of one daughter cell obtained during mitosis, while the other daughter cells enlarge and differentiate. An example of arithmetic growth is the elongation of roots. Notice that only one cell keeps dividing, while the resulting daughter cells don't divide but only differentiate. If we plot a graph of length against time for the growth of a root, we will obtain a linear graph. The rate of growth is given as the gradient of the graph. Geometric growth refers to the division of both daughter cells obtained after each mitotic division. This leads to an exponential increase in the number of cells. This type of growth is seen in the early stages of development, that is, development of a zygote to an embryo. A graph of the geometric growth is of a sigmoid nature. The curve depicts the different phases of growth in living organisms under natural conditions. Growth is lower in the initial phase, called the lag phase. Here, the number of dividing cells is small, and the cells are not yet adapted to the new environment. Then the growth is rapid at the exponential or log phase. Here, there are more cells dividing. The cells have adapted to the new environment, and there is sufficient food and other factors such as oxygen. Also, the rate of cell division is higher than that of cell death. Then the growth starts to slow down at the decelerating phase. Here, most cells have differentiated, and so only a few cells are dividing. There is also a shortage of nutrients and other factors such as oxygen. There is unlimited space, and also there is buildup of metabolic waste. Then finally, net growth ceases altogether in the plateau or stationary phase. Here, the rate of cell division equals to the rate of cell death, and nearly all the cells have differentiated. Growth and development in plants. Plants exhibit unlimited growth. That is, they retain their capacity to grow throughout their lifetime, from germination to death or senescence. Seed structure and germination. Previously, we said that in biology, a fruit is a fertilized ovary with seeds. Based on the number of cotyledons, seeds of flowering plants are classified as dicotyledonous and monocotyledonous. A seed with 2 cotyledons is called a dicotyledonous, and one with a single cotyledon is called a monocotyledonous seed. In a dicotyledonous seed, the outermost covering is the seed coat, and it's made up of 2 layers, the outer testa and the inner tegmen. The seed also has a scar on its outer surface called the hilum, which was the point of attachment on the fruit. Above the hilum is the micropyle, a small hole in the outer coat of the seed. It is through the micropyle that a pollen tube entered the ovule. Inside the seed coat lies the embryo, which comprises of an embryonal axis and 2 cotyledons. At the 2 ends of the embryonal axis lies the plumule and the radicle. The cotyledons are often fleshy and store reserve food materials. In some seeds, such as castor, the developing embryo is surrounded by a nutritive tissue called the endosperm, which stores food and helps the embryo grow. These are called endospermic seeds. Like dicots, the outside covering of the monocot seed is also known as a seed coat. For instance, the maize seed has a membranous seed coat that is usually fused with the fruit wall. And just like in dicots, monocot seeds can be endospermic like in maize or non-endospermic. The endosperm of the maize is bulky and stores food, while the embryo is tiny and is placed in a groove on one end of the endosperm. The embryo is separated from the endosperm by a proteinous layer known as the aleurone layer and is made up of a single, large cotyledon called the scutellum. The embryo has a short axis with a plumule and a radicle. The plumule is enclosed by a sheath called the coleoptile, and the radicle by a sheath called the coleorhiza. The process by which a seed develops into a seedling is called germination. First, the seed absorbs water through the micropyle, causing it to swell in a process called imbibition. The water dissolves the stored food, which is oxidized to release energy that powers the cell division and hence the growth of the embryo. The embryo enlarges and breaks the seed coat. The root emerges out first and grows downwards. And later, the shoot emerges, which grows upwards. The cotyledon leaves turn green on exposure to light and therefore start to photosynthesize. This increases the speed of growth and more leaves are produced. The conditions necessary for germination include water, which dissolves the stored food and therefore provides a medium for enzyme activities. Oxygen is required for respiration to provide energy for embryo growth. Temperature activates enzymes. Enzymes including diastase, proteases, and lipases are required to break down the respective food materials. Hormones such as gibberellins and cytokinins are required as growth stimulators. Also, the seed has to be viable, that is, the embryo must be alive. There are two types of germinations, epigel and hypogel. Epigel germination occurs in dicots. During this germination, the hypocotyl, that is the part between the cotyledon and the radicle, curves and pushes upwards through the soil, carrying with it the two cotyledons, which turn green and leafy once exposed to sunlight. Hypogel germination occurs in monocots. During this germination, the epicotyl, that is the part between the cotyledon and the plumule, elongates and the plumule grows out of the coleoptile while the cotyledon remains underground. Seed dormancy. Now, the embryo of a dry, fully developed seed usually passes through a period of rest after ripening period. During this time, the seed performs all its physiological processes very slowly and uses up very little food. This is called the period of dormancy. Even if all the favorable environmental conditions for germination are provided to the seed will not germinate. This is due to the fact that the seed embryo may need to undergo further development before germination. Dormancy has several advantages to the seed. It provides the seed with enough time for dispersal so that they can germinate in a suitable environment. It also enables the seed to survive during adverse environmental conditions without depleting their food reserves. The embryo has enough time to develop until favorable conditions are available. Factors that may cause dormancy include a mature embryo that still needs more time to develop, germination inhibition by chemicals, lack of or low concentration of hormones like gibberellins, hard and impermeable seed coats that prevent entry of air and water, or freezing of seeds during winter. Next, primary and secondary growth. Now, plants increase in both height and girth or width. Plants' growth occurs due to the presence of meristematic tissues which have the ability to divide. The cells are similar, generally small, and are cubical. Depending on the region they are present in, meristematic tissues are classified as apical, lateral, and intercalary. The tissues that are responsible for increase in length are the apical meristems. They are located at the tips of stems and roots. This kind of growth is called primary growth. The regions of growth in a plant root include the zone of division, elongation, and maturation. The zone of division contains rapidly dividing cells of meristem. These cells are characterized by a rich protoplasm or cytoplasm, a large nuclei, and a thin primary cell wall with more plasmodesmatal openings. The zone of elongation lies next to the meristematic tissues. This region is characterized by enlarged cells, deposition of a new cell wall, a thin layer of cytoplasm, a large vacuole, and a tiny nucleus. Next is the zone of maturation. This zone is characterized by mature cells that have reached maximum size in terms of cell wall thickening and cytoplasm. Now, secondary growth. The tissues responsible for increasing the diameter of the plant's body is the lateral meristem, located on the lateral position of the roots and stems. This is called the vascular cambium. At the beginning of the year, the vascular cambium is dormant. It then becomes active, swells, and undergoes its first division, forming the xylem mother cells. The cambium then divides again, forming another xylem mother cells, while the first xylem mother cells begin to differentiate. In the third division, the cambium divides, producing the phloem mother cells, while the old phloem begins to be pushed and crushed on the outside. Next, the cambium undergoes multiplicative division, allowing for growth in circumference. In the fourth division, the cambium divides into another xylem mother cells, while the existing cells continue to differentiate. In the fifth division, another xylem mother cells form, while the cells formed in the first division have become fully differentiated. In the sixth division, growth begins to slow down, and the xylem mother cells that are formed become latewood. The seventh division shows the cambium dividing, forming phloem mother cells, while a layer of the outer bark is shed. In the eighth division, the final layer of the xylem mother cells are formed while the previous phloem matures. Finally, the cambium cell division finishes for the year, while the existing xylem mother cells differentiate into latewood or earlywood, and the cambium becomes dormant again. Now, plants development. Plants development basically involves maturation and differentiation. We've seen that the root meristem, cambium, and the shoot meristem add new cells. These new cells undergo structural changes for specific functions. This is called differentiation. For instance, some of the meristematic cells of the procambium lose their protoplasm and develop strong, elastic secondary cell walls to form vascular tissue. This tissue helps in the transportation of water and food throughout the plant. In addition, the procambium cells differentiate into parenchyma cells, which act as supportive tissue. Differentiation in plants is open, similar to growth, because cells formed from the same meristem differentiate into different structures at maturity. The final structure of a cell at maturity is determined by its position on the plant. For instance, cells close to the apical meristem differentiate into root cap cells, while cells at the periphery differentiate into the epidermis. Next is plant growth regulation. In the life cycle of plants, cell growth and differentiation are interrelated and controlled by intrinsic and extrinsic factors. Extrinsic factors include light, temperature, water availability, oxygen, and nutrition. The increase in cell size is due to absorption of water. Water also acts as a medium for chemical reactions required for growth. Oxygen is required for respiration to release energy. Macro and micronutrients are needed as energy sources and synthesis of cellular contents such as protoplasm. In addition, plants need an optimum temperature for enzyme activities. The growth of plants depends on light because they manufacture food by photosynthesis. Intrinsic factors include plant growth regulators called PGR. These regulators are chemical compounds and are also called plant hormones or phytochromes. They include auxins, gibberellins, cytokinin, abscisic acid, and ethylene. They are divided into two groups: plant growth promoters and plant growth inhibitors. Plant growth promoters enhance plant growth activities such as cell division, cell enlargement, flowering, fruiting, and seed formation. Auxin, gibberellin, and cytokinin are plant growth promoters. Plant growth inhibitors respond to wounds and stress from biotic and abiotic factors by inhibiting growth. Abscission, that is the falling of leaves due to a scar, and dormancy, that is a state of inactivity, are due to the actions of plant growth inhibitors. Abscisic acid acts as a plant growth inhibitor, while ethylene can act both as a promoter and an inhibitor. Auxin is a generic name for all natural and synthetic compounds with definite growth regulating feature. The most common auxin is indole acetic acid produced at the apex of the stems and roots. Auxins are used in crop propagation because they cause rooting in stem cuttings. They also induce flowering, as in pineapples, prevent falling of young leaves and fruits, and promote abscission of mature leaves and fruits. Auxins are used in parthenocapy, that is the formation of seedless fruits such as tomatoes from unfertilized flowers. They cause apical dominance in higher plants, that is, they inhibit the growth of lateral buds. This is why the shoot tips of tea plants are cut to initiate growth of lateral buds. A synthetic auxin, 2,4-dichlorophenoxyacetic acid, commonly called 2,4-D, is used as a herbicide. It uses the physical differences between leaves of monocots and dicots to selectively kill dicots by inducing distorted growth and excessive respiration. Gibberellins or gibberellic acid are extracted from fungi and higher plants. They cause increased lengthening of the plant's axis. They are therefore used in lengthening of grape stalks and sugarcane stems to increase the yields. They also improve the fruit shape by causing elongation, as in apples. They delay senescence or aging of fruits, and so are used commercially to extend the market period by retaining fruits on trees longer. However, they also hasten maturity of seeds and are hence used to obtain early seed production from juvenile conifers. Gibberellins are also important in fruit formation by inducing growth of ovaries into fruits after fertilization, as well as inducing parthenocapy. They promote formation of lateral branches by breaking dormancy in buds. They reduce leaf fall by retarding formation of abscission layer and also break seed dormancy, hence stimulate germination. Cytokinin plays an important role in cytokinesis during cell division. Natural cytokinins can be found at the root apex, shoot apex, and fruits. They promote the growth of lateral and adventitious shoots by preventing apical dominance. They also delay senescence or aging of leaves. Together with auxins, cytokinins stimulate cell division and therefore bring about rapid growth of roots, leaves, and buds. They promote flowering and breaking seed dormancy. Ethylene is present in plants in gaseous form. It is synthesized in the plant organs that undergo senescence, such as ripened fruits. Commercially, it is available in aqueous form, ethyphone, which can easily be absorbed by plants. Ethylene gas is released from ethaphone, which accelerates the ripening of fruits by enhancing the rate of respiration. It is therefore used in the artificial ripening of tomatoes and apples. It promotes the abscission of leaves and flowers, and thus favors the thinning of cotton, walnut, and cherry. Ethylene also breaks seed and bud dormancy and is hence used to enhance the germination of peanut seeds and potato tubers. It also helps in lengthening of internodes in rice plants and thus preventing the shoot from being submerged in water. It promotes roots and root hair formation and thus helps the plant to increase their absorption service. It initiates flowering in some plants such as mangoes, pineapples, and cucumber, thus increasing their yield. Abscisic acid is a plant growth inhibitor that regulates abscission and dormancy in plants. It helps plants to cope with stress and is hence known as a stress hormone. In cases of unfavorable environmental condition, it inhibits germination in seeds. In drought conditions, it closes the stomata, thus preventing transpiration and hence conserve water. In fact, the effects of abscisic acid are antagonistic to those of gibberellins. Another plant growth regulator is called florigen, which is synthesized in the plant's leaves. When the plants are subjected to light, this hormone moves to the shoot tips where it induces flowering. And now growth and development in animals. Just like plants, higher animals' bodies are made up of different types of cells, like bone cells, cartilage cells, blood cells, muscle cells, nerve cells, and many more. And yet, this multicellular organism began as a single cell, a fertilized egg or a zygote. A little more than a day after its formation, the zygote starts moving down the fallopian tube. During this movement, the zygote also undergoes mitotic divisions without increasing in size, but the number of cells double with each division. This division is termed as cleavage, and the resultant daughter cells are called blastomeres. About 3 days after fertilization, the zygote contains nearly 8 to 16 cells and is called a morula. Over a period of 2 days, the morula descends down into the uterus and transforms itself into a blastocyst, a hollow ball composed of about 100 blastomeres arranged in 2 layers, an outer layer called trophoblast and an inner layer called the inner cell mass. The embryo then attaches itself to the uterus, with the trophoblast growing outwards and penetrates the endometrial lining of the uterus. Finger-like projections called chorionic villi grow and interlock with the uterine tissues to form the placenta, which is a site of exchange of materials between the mother and the developing embryo. The inner cell mass moves downwards and differentiates to form an embryonic disc consisting of 3 germ layers: the outer ectoderm, middle mesoderm, and inner endoderm. The term germ indicates that each layer gives rise to tissues. The ectoderm, for instance, gives rise to the nervous system and the skin, whereas the mesoderm develops into muscles and organs such as the heart and kidneys. The endoderm gives rise to the entire digestive system. As the embryonic disc grows, the cells on one side of the disc grow faster than those on the other side, causing the disc to roll and form a cylindrical embryo. Initially, this embryo is attached to the placenta, but as it starts to develop, it moves away from the placenta and remains connected to it via the umbilical cord. The embryo stays in the uterus for 9 months, which is called the gestation period. During this period, the embryo undergoes several developments. By the end of 1 month, the embryo develops a heart. The end of the 2nd month marks the transition of an embryo to a fetus with developing fetal limbs and digits. And by the end of the 3rd month, most of the organs are already formed. Another 4 months and the fetus is rapidly gaining weight and its bones get stronger. By the beginning of the 9th month, the fetus is fully developed and assumes the position it will take during delivery. At birth, the fetus has all the organs that an adult has, and so humans and other higher animals undergo direct development, unlike amphibians and arthropods. And finally, growth and development in insects. Unlike in humans who undergo direct development, arthropods, like insects, go through a series of changes to become an adult. Each of these stages is very different from the others. This process of transformation from an immature form to an adult through distinct stages is known as metamorphosis. Metamorphosis can be complete, like in a bee, or incomplete, like in a grasshopper. Let's look at the life cycle of a butterfly. When you look at a butterfly, it is hard to imagine that what you are seeing is the product of a complex growth process that sometimes biology itself sounds like magic. First, the adult butterfly lays eggs on a leaf. Inside the egg, the larva starts to develop. And once fully developed, it hatches by feeding on its own shell. In fact, once it is out, the larva, also called a caterpillar, completely feeds on the remaining shell. The larva stage is a very active stage. The caterpillar feeds on foliage and grows rapidly in size with several molts to shed off the exoskeleton or the cuticle. It is the most destructive and devastating stage to crops. When the time comes, the larva will look for a shaded and secure place to attach itself, ready for the next stage of life. It securely attaches itself to a surface. The larva is then encased in a puparium and enters the pupa stage. This is a non-feeding stage. Although many people describe it as an inactive stage, that is only on what we see from outside. But inside, an intricate transformation is taking place. It has to get rid of the caterpillar tissues because they won't work in an adult. In fact, the cells themselves disappear, but their components are recycled and turned into some kind of a fluid out of which the adult structures are built. It is a very carefully engineered process. Once completely formed, a new organism, an adult butterfly, emerges from the puparium. This whole process of metamorphosis is controlled by hormones. The hormones are produced by three glands, namely the neurosecretory cells in the brain ganglia, a pair of corpora alata located in the mandibular segment, and prothoracic glands in the thorax. During the larval stages of the insect, the corpora alata produces juvenile hormone. This leads to the formation of larval cuticle. Therefore, molting does not go beyond the larval stage. When the larva matures, the corpus alatum disintegrates. At this time, the neurosecretory cells stimulate the prothoracic glands to produce molting hormone or ecdysone. Ecdysone is responsible for molting in insects, leading to the laying of adult cuticle. And finally, growth pattern in arthropods. Now, whereas growth in plants and animals is smooth and continuous, growth in arthropods is intermittent. It occurs in intervals, and that is because the chitinous exoskeleton limits the expansion of the insect tissues. In order to grow, arthropods often shed off the exoskeleton time and again to allow for expansion of tissues. When most people see an arthropod outer shell, they think the animal had died. But no, these are exoskeletons of arthropods that were shed off. This shedding off of the exoskeleton is called molting or ecdysis. While cell division continues to take place inside the insect body, they don't expand. To do that, the exoskeleton needs to be shed off. The new arthropod then inhales air to expand the cells. A waxy cuticle then develops over the new exoskeleton to make it waterproof. The molting process begins when the cuticular epidermal cells are stimulated by the molting hormone. The cells undergo mitosis. This is the period of growth to form a new larger cuticle for the next instar. The existing cuticle separates from the epidermal cells in a process called apolysis. The resulting ecdysal space is filled with a gel that contains inactive molting enzymes. A new outer epicutical layer of cuticulin is secreted. This layer protects the epidermal cells and the newly forming cuticle from digestion by the enzymes in the molting gel, which is then activated and becomes a fluid. The activated enzymes begin to digest the old endocuticle. This continues until it reaches the old exocuticle, which is resistant to enzyme action. The remaining molting fluid is absorbed. New layers of endo and exocuticle are deposited by the epidermal cells. Just before molting, a cement layer is released by the dermal gland. Notice the old cuticle is smaller than the new one, and this is because there has been growth of the epidermal cells. Molting or ecdysis occurs when the old exocuticle is sloughed off. The shed of cuticle is called exuvium. During the time between the molds, new endocuticle is deposited continuously and the cycle starts over again for the next mold.